in the 5 kingdom classification, the first kingdom is Kingdom Monera. This Kingdom Monera includes prokaryotic, unicellular organisms. This Monera organisms will show some special characteristic features, that they lack organized nucleus, or we can say true nucleus is absent. True nucleus or definite nucleus is absent. And here it is represented in the form of nucleoid or genophore. Then they have 70s type of ribosomes, and majority of them, they may show autotrophic nutrition. At the same time, some will exhibit the heterotrophic type of nutrition also. And when it comes to the cell wall nature, so it is generally non-cellulosic in its nature, non-cellulosic cell wall, which is made up of polysaccharide, different types of polysaccharides and amino acids. So these are the basic features that can be observed in the Monera group. And the Kingdom Monera have been further classified into subdivisions. So of them, first one is Archaebacteria, Eubacteria, Mycoplasmas, and Actinomycetes. So these are the groups that have been included under Monera kingdom. So let us discuss the characteristic features of individual group of organisms which have been kept under Monera. Starting with first one, Archaebacteria. So actually the group of organisms which have been kept under Monera are considered to be derived from the early livestock, that is progenotes. They have been derived from early livestock, that is progenotes, and exhibiting the prokaryotic nature. So now let us come to the first group which is kept and which is included under Monera, that is Archaebacteria. Archaebacteria are considered as a primitive group of bacteria, and Archaebacteria have the special feature that it have the ability to survive in harsh habitats also, and of course it is present in majority of the habitats, but it have the ability to adjust to the harsh habitats. And apart from this one, here why it have that ability to survive in such type of harsh habitats means the composition of the cell wall, that is very much important, which is helping in adaptation to such type of harsh habitats. So here the cell wall is not having peptidoglycans. Or absent. Peptidoglycans are absent, and murines are also absent, which is actually the characteristic feature of individuals which are kept under Monera. Whereas here in place of these peptidoglycans, so this cell wall is made up of pseudomurines. Cell wall is made up of pseudomurines, and apart from these pseudomurines, they have special lipids or branched chain lipids in their cell wall. So because of this branched chain lipids or the special lipids, they are helping in the survival of in the harsh habitats.